First of all, I would like to thank the organizers, Department of Botany, Giriraj Government College in collaboration with Council for Green Revolution, Hyderabad, and Department of Botany, Telangana University, Nizamabad, for giving me this opportunity. I really, it's my pleasure as well. And as already been stated, my talk is entitled Symbiotic Effect on Mycorrhizae on Plants. Now, we just wanted to start what is mycorrhiza. We all know that it has a kind of a mutual symbiotic association between a fungus and a plant. And it also refers to a role of fungus in the plant's rhizosphere and its root system. So when we say rhizosphere, it is around the root is there in a plant, and surrounding of the soil, we say it is rhizosphere, and on the surface of the root, we say rhizoplane. So rhizoplane as well as rhizosphere, it really plays a big role with here to having a mutual association with fungus and the plant. So mycorrhiza, it plays an important role in the plant nutrition, soil biology, and soil chemistry. We can also say it's a soil biota. Here I have shown the figure where we can see that when there is a mycorrhization in the plant, it has a very good proliferation of rooting system with a very fine rooting system here. And when there is no mycorrhization, that is a control plant, we can see that in the right, there is no mycorrhization. So we know from the history that mycorrhiza, it has started in 1880. The term has been coined by A.B. Frank. And since it is in a Latin word, so how that the word has become mycorrhiza. So there are various types of mycorrhiza. Firstly, we can say it is ectomycorrhiza. Here in the picture, in the line drawing, we can see that it is the ectomycorrhization, where the root or the mantle of the root is been surrounded by the hair-like structures, which is mycorrhizae. In the next, endomycorrhiza. When we say endomycorrhiza, it is within the cell wall, and it will penetrate in the cell wall and invaginate the cell membrane. So this is where the place you can find the mycorrhiza. So it is known as endomycorrhiza. So when we say it is ectendomycorrhiza, that is the third type, it belongs to the same Basidiomycotina and covers both gymnosperms and angiosperms. That is the class of the plants. So it is known as ectendomycorrhiza because it is having in the both family it is covering. So we say it is ectendomycorrhiza. And generally it shows extensively the growth in within the intercellular penetration. So it will begin a formation of heartic neck. It is a kind of a network of the mycorrhiza, and which it grows behind the apical meristem of the growing root. And this heartic net, it penetrates between the epidermal and outer cortical cells and later extends to the inner cortex region. So when we talk of applications of mycorrhiza, we say in a singular form, mycorrhizae, it is in the plural form. And it increases nutrient uptakes of the plant from the soil. So generally when we say it is the nutrients, it is in the mineralized form, that is the phosphorus nutrition and other elements like nitrogen, potassium, calcium, manganese, zinc, copper, sulfur, boron, molybdenum, iron and manganese and chloride, etc. And it will also increase the mycorrhization. It increase in the diversity of the plant also, and it produces uniform seedling, and it has a significant role in nutrient cycle, recycling also, and it is more tolerant to adverse soil chemical, and it is constraint which limit the crop production. It helps in the increase of plant resistance to various diseases and drought and salinity conditions, and also it helps to stop the pathogenesis of the plants. and it stimulates the growth of beneficial microorganisms, which will help for the promotional effect like PGPRs, which will help the plants for the growth and promotion of the plants and improving the biomass, etc. And it will also improve and help in soil structure, that is conditioning of the soil, and it stabilize soil aggregate and hyphal polysaccharides that bind and aggregate soil particles, right? So this will help the conditioning of the soil. It increases absorption of phosphate by crops or some other medicinal plants, flowering plants, and forestry plants, etc. It will help to uptake zinc, and it also increases its uh, um, availability in the root system, and it also helps in uptaking water from the soil with the capillary action. And it is believed that from the root system of the plants, it almost goes to 100,000 feet away from the rooting system. This mycorrhization can help in uptaking the nutrients and water to the plant. It increases uptake of sulfur from the soil. This has been already been said also. And also concentration of cytokinins and chloroplasts in plants is increased. And it will also protect during the stress conditions in salinity, in the harsh conditions like high pH level, and in drought condition, etc. So in short, the types of mycorrhizal fungi, it has 7 classes here. You can see 7 types: Ectomycorrhiza, arbuscular mycorrhiza, ericoid mycorrhiza, arbutoid mycorrhiza, monotropoid mycorrhiza, ectoendomycorrhiza, and orchidaceae mycorrhiza. Here you can see in the picture that how these 7 types of mycorrhization it occurs within the rooting system of the plants. Of course, it is in the line drawing. And it, as I have already told you, it has a very vital, big role in overcoming the drought stress condition. Like when it is, there is a vitrification in the glass formation, and protection of membrane is being taken if the sugar is very high. If protein is very high, then dendrites and heat shock proteins, these can be also maintained by the mycorrhizas, and antioxidants and various types of enzymes that can be also maintained by the mycorrhiza. And there are free radical scavenging processes also helped by the use of mycorrhizas. And osmoregulation, that is the water uptake, is also being regulated by the mycorrhiza. So in totality, almost it has been observed that 90% of plants are colonized by mycorrhiza, and mostly these falls under economically important crops. So when we talk of beneficial part of mycorrhiza, we will divide into 2 parts, like for the beneficial for the plants itself, and the beneficial portion that is for the fungus itself. When you talk of plants, that is connection between root and soil around the environment we are talking of, that is the rhizosphere part, and it helps the growth. It helps as a growth promoter for the plants, and it will help in the nutrient uptake, increasing the surface area, increasing mobilizing unavailable nutrients like rock phosphate, which is not easily being taken by the plants itself. And obviously there are some microorganisms like other bacteria also help in chelating this rock phosphorus, but however this mycorrhiza can also help in chelating the rock phosphorus, which can be easily taken up by the rooting system of the plant. And excreting chelating compounds, or sometimes ectoenzymes, is also being transferred by the mycorrhiza to the plants. When you talk of benefits to the plants only, fungus only, it helps to transfer metabolites to the from the plant to the fungi. It will take up, And plants transfer almost 10 to 20% of carbon to the fungi, because as we know, most of the fungi, it lacks in chlorophylls also. Like some of them are also mushrooms, where this mycorrhization helps to uptake the nutrient materials to the fungus. And nowadays, there is a kind of a arbuscular mycorrhiza like fungus, and mostly the arbuscular mycorrhiza, these are known as glomus mosaic, and there are so many types of fungus which can grow within the root, but their sporulation can be seen in the soil as well, but it cannot be cultured in the lab alone. But very surprisingly, there is arbuscular mycorrhiza like fungus. We say like because it is all the characteristics is mimicking with the arbuscular mycorrhiza like AMF fungi. But this Piriforma spora indica, it is easily culturable in the lab condition also. That is why we say it is AMF like fungi. And nowadays, this Piriforma spora indica, it has been known as Serendipita indica, which is also the same, only the naming is different. And here we can see that this is the root section. Staining has been done by the trypan blue, where you can see that in the section of the root, you can see that within the cell wall, we can see that there is a spores growing within the root cells, right? And these has been magnified here to the extreme right lower side. This has been magnified so let you see that how the spores are looking like. It looks like the shape of the spore is like a pear shaped. So because of this, how the name has been coined as Piriforma spora indica. This was first isolated in association with spore of Glomus mosaic from this is the AMF fungi, Glomus mosaic, and it was associated with this fungi. So this has been isolated from the rhizosphere of two shrubs in the Indian Thar desert, that is the northwestern Rajasthan, in 1990. And the paper was published for the first time by Virma et al. In 1998. So soon after, the fungus was characterized as a mutualistic endophyte because of his ability to colonize plant roots without causing any visible disease symptoms in roots or shoots. So instead of it confers a strong growth promotion and induces disease resistance to microbial pathogens. So most of the laboratories within the world, almost 23 laboratories are working in this fungus nowadays. So molecular phylogenetic analysis have revealed that P. indica belongs to the Basidiomycetes, that is the order of Sebaceales. So it is also known as Sebaceales now. The salient features of AMF like fungi, that is the P. indica, you can see that it is axenically culturable on synthetic media. So this is a very different drastic characteristic from the AMF fungi, which is not culturable, but of course you can grow this fungus AMF fungi within the plant root in the laboratory as well as with the plant in the greenhouse and also in the ex situ and in situ condition. But this P. indica, you can just take out the spore and culture in the synthetic media in a petri dish. This is the difference that has been observed from the AMF fungi, and it has no clamp connection, and there is anastomosis is occurring frequently. Hypha aggregation is often observed, no hyphal knots, and simple septum with dolichopores and continuous straight parenthesomes are being seen. And chlamydospores are a little bit smaller in compared to AMF like fungi. AMF fungi. That is 16 to 25 micrometer in length and 10 to 17 micrometer in wide. And then 8 to 25 nuclei spores has been seen, which I have also shown in the trypan blue staining, where you can see the nuclei within the spore. So what are the functional similarities with AM fungi? So when we look through it, we can see that broad and diverse host spectrum as similar to AMF fungi. Hyphae extramatrical and intracellular is being seen within the root region. And hyphae never invade the endodermis layer. And chlamydospores in soil and within cortical tissues is being seen. Sexual stages till date is not seen. Positive phytopromotional effects on tested hosts is being seen. Phosphorus mobilizer, phosphorus transporter, tool for biological hardening of micropropagated plantlets is being seen, and it's doing really good. And potent biological control agent against root pathogens has been also tested. And thus, this Piriforma spora indica, besides the biological hardening, that its protoplast can be also applied and for the fundamental researches and genetic transformations. And when comparing 18SR DNA sequences that is obtained after the PCR, it is showing a kind of fungus apparently that occurs in the rhizosphere, which is almost similar, which has been found in Pakistan and Philippines. That is with the similar to rice plants and some few grasses. But however, it is not the exact findings is being seen, but it is similar. The host spectrum of Piriforma spora is very much alike like AMF fungi. That's what I'm saying still, that it is known as AMF like fungi, where we can see here similar to AM fungi, this has also 90% of the terrestrial plants colonization, and specifically mostly economically important plants. And it is also shown that it is well inoculated in medicinal and forest and ornamental plants, in horticultures, in floricultures, viticultures, etc. It has been successfully tested. And one of the striking differences is that unlike AM fungi, it has a host range of Piriforma spora indica with terrestrial orchids, where this AMF fungi is not colonizing in the terrestrial orchids, but this Piriforma spora indica is um, um, it is colonizing terrestrial orchids. So nowadays, since it's showing as a um, biofertilizer, it's acting as a bioprotector and as a also like a herbicide effect or bioprotecting effect. So right now, it is also known as a root tonic. So this is the exact form of electron microscopy of Piriforma spora indica hyphal and the spores, where you can see with this white arrow, it's been showing this pear-shaped spores has been seen. That is how this name has been coined. And there is a hyphal coil here in the middle, you can see. And that is when it has been magnified, the hyphae has been magnified, you can see that there is a kind of a funnel type of apristoria is being seen. And this apristoria is very much helpful in transporting the nutrients to the plant. Now, when we talk of mycorrhization, how it can be done? It seems simple, it can be done. So in the laboratory, in the in situ or within the greenhouse, we can do this mycorrhization, and we can do the mass propagation of this Piriforma spora indica. Here you can see that soil inoculum is there, obviously, like similar as with other AMF fungi. And you take one flower pot that is semi-sterilized. We say semi-sterilized because since we are using with the soil and sand here, now, this cannot be completely sterilized as you do with some other bacterial media, right? So soil and sand is been taken as 1:3 composition, and it has been successively, it has been autoclaved in alternate days, heating cooling system. Then it has been the sand is well washed with the acid, and then with lots of water you wash off all the acids remaining. So the crevices in the sand, there is no other natural mycorrhiza left or any other organisms left. So you make sterilized the substrate. Now you say 1:3 composition of sand and soil composition. Then what you do is one layer you put this lower layer, the substratum. Then in the middle layer you put the inoculum, that is a soil inoculum containing the Piriforma spora indica. Then again on the top layer you put the substratum. Now you go for the planting of the seedlings. Here has been the corn maize seedling has been seen, where you can implant this seed within the layer of the inoculum. This is how you do the mycorrhization. And the second style you can do is, the technique you do is, you take the culture, as I have told you that it can be easily cultured in the laboratory, in the synthetic media, that is a MM media, that is a mineral media, or even in potato dextrose simple medium you can do it, because this is a fungus, right? So it can easily grow in the synthetic media, where you can similarly grow the other fungus. After growing this, about 5 to 7 days it will grow radially and it will cover the whole media when you place a plug of inoculum in at the center of the culture media. So you make a, with a sterile borer, you can make a punch hole. Then that uh, or cock borer can be also taken, and that plug is you take as in the substratum, as before in the earlier slide I have told you, make the substratum as 1:3 sand soil composition. Then you put the plug about 2 centimeter deep, right? Then you plant a seedling, or you can also put the maize seed within it and then cover with a substratum and wait for 15 to 3 months. Then this is how the proliferation or mycorrhization can be produced. This can be also similarly worked for the AM fungi. Since this is an AMF like fungi, you can also do the similar technique. So exenic culture shows that these are the spores of the Piriforma spora indica on carrot root organ culture, where it can be also done in the laboratory condition. You can get lots of spores in this case, in this way. So for identification, you can easily collect the spores and then take out the protein out of that, and then get the antigen. So you can go for the immunization in rabbit or mice to get the antibodies. So how that way also in serological also by using serological technique, you can also characterize the fungus. So in the lower side you can see the spores sporulating in the root organ culture, that is in the carrot root organ culture. And this is a biological hardening that has been done with the Withania somnifera, which is a medicinal plant of course. And here you can see with the mycorrhization plus, that is the mycorrhization minus means that is the control plant without mycorrhiza, that is again Piriforma spora indica, that is AMF like fungus I am talking about. And you can see that there is a proliferation of leaves. The plant is very nice and very dark green in color, and lots of biomass has been produced. And later it's also been shown that fruiting and flowering and fruiting bodies is also more in case of mycorrhization in compared to non-mycorrhization. So as I have told you, in the serological identification technique, it's not only that for the pathogenic organisms, for the human disease that you can use, or the plant disease, or in animal disease that you can produce antibodies against it, but for the taxonomy also you can raise antibodies and then use for the identification of the uh, that particular fungus. Here we are talking of Piriforma spora indica, where antigen is being taken from the spores of Piriforma indica by crushing it in a buffer, and then you take this, obviously the protein, it is the protein. We all know that when you take the antigens, it can be protein or it can be uh, some kind of peptides you can take, right? And the antibodies is raised against it, and there is secondary antibody, that is the enzyme level HRPO for the color formation, sorry, This has been done in the ELISA reader, in a, and then you can see that there is antigen and antibody complexes being formed with the help of secondary enzyme labeled secondary antibody. There is a fluorescence is being seen, and this reading has been taken in the ELISA reader. And similarly, as earlier also, one of the previous speaker has also talked about Western blot technique also. For the here, this is the dot blot is also done for the identification of the antigen and antibody that is being raised against Piriformaspora indica for identification of the particular strain, that is the Piriformaspora indica. And when it has been, this is just the figure to show you that how it can be cultured in the synthetic media. This is media, that is the minimum media, where a plug of agar containing the spore of Piriformaspora indica is placed, and after 7 days it has a radial growth growing. And similarly, the other Sebaceanil, that is Sebacina vermifera, which is almost similar to Piriformaspora indica. This Sebacina has been identified in the European countries in 1960s, and this Piriformaspora indica, it has been identified in the in India in 1998, right? So it has both of it. It is in the same family, almost having the same type of growth, but however these two are different. So I was talking about Ectomycorrhiza also. These are all the Ectomycorrhizas. The name is given here, like Trichoderma and then Amanita, etc. So these are also culturable in synthetic media, similar as Piriformaspora indica. Only that different is Piriformaspora indica is AMF-like fungi, but these are the Ectomycorrhiza, because these does not colonize the cortical region of the root. It only develops the mantle around the root only. So when Piriformaspora indica is being taken, this A is controlled without any mycorrhiza, A controlled without any mycorrhiza. B is for Piriformaspora indica. You can see the proliferation of the root here, plantation here, shoot I mean to say. And this C is for Sebacina. So similarly here, this is the control. This is Piriformaspora indica, this is Sebacina. These are Glomus mosi and another Glomus species. So you can see here that comparing to another AMF fungi, and this Sebacina is also AMF-like fungi, and P indica, this is also AMF-like fungi. It's showing the higher shooting and rooting effect. So these are the maize plants, and this rooting system is also from the maize plant, where control there is no proliferation of the rooting system. In P indica, you can see that their rooting system are proliferated and it has become lower and hairy and more in biomass. Similarly with Vermifera also, but in this is AMF fungi, you can see Glomus mosi, it's a little lesser than in compared to P indica. When we talk of biomass, this is grown in the broth culture, and this is from the 5 days, 7 days and 10 days replica, and we see that similar method has been followed for the Sebacina, that is the another kind of AMF-like fungi which is found in the European countries, and P indica, that is in the India, which is been found. We can see that almost similar it is, but however P indica is a little bit higher in rate in compared to Sebacina. So this is the cytochemical staining by trypan blue. You can see here, this to the left is all P indica, and to the right is all in Sebacina vermifera sensu, which is almost similar. And through microscopy tests, it's difficult to identify. It looks same. So in the end, I can see that I can say that there is lots of research area has been opened due to this experiments previously done by, that is myself also. I did my PhD in the same fungus from Jawaharlal University under Professor Dr. Ajit Verma, who is my mentor. And similarly, there are so many other students has already done the same work same similar work under the Piriforma spora indica, but still there are yet to do long way to go. But however, it in the end I can give recommendation as such that it acts as a, this is the Piriforma spora indica spores in the trypan blue staining. It acts as a plant promoter, as a biofertilizer, as a bioregulator, as a bioherbicide, and immunomodulator and phytoremediator. Biocontrol against insects and pathogens has been also seen. It has a stress tolerance uh, effect also, and like salinity, temperature, etc., and drought condition also. And it acts as a stimulator with PGPRs, that is plant growth promoting rhizobacteria, and it acts as an antioxidants and drug enhancer. There is yet to do lots of work in this area, and it also enhances anti-aging agents also. So yet to go far ahead for the in experiments and researches. And it also acts and promotes anti-cancer drugs, and yet to go more to come. So with this, I would like to end my talk here, and thank you for listening.